webinar titled Musing of a Scientist, Incremental Steps Towards Commercialization. This webinar promises to be So it gives me an immense pleasure to extend a heartfelt welcome to each one of you to our first HBNI monthly alumni webinar titled Musing of a Scientist, Incremental Steps Towards Commercialization. This webinar promises to be an enlightening experience for all of us, particularly to our students seeking guidance in their placement endeavors. I welcome today's HBNI alumni speaker, Dr. Nikhil Sangith, Director, Exact Diagnostic Private Limited. I extend my heartfelt gratitude to Dr. Sangith for generously sharing his time and expertise with us today. Now I'll give a brief introduction to Dr. Nikhil Sangith. He obtained his graduation from PhD College of Arts and Science, Bharatihar University, Coimbatore in 2006 and post-graduation degree in biotechnology from Amrita Vishwa Vidyapitham, Kollam, Kerala in 2008, and later received a PhD in Life Sciences from Advanced Center for Treatment, Research and Education in Cancer, ACTREC, ACI of HBNI in 2015, under supervision of Dr. Prasanna Venkataraman. After PhD, he was postdoctoral research fellow in Tata Institute of Fundamental Research, TIFR, during 2015 to 2016, and a research scientist in Department of Biotechnology, Anna University, 2016 to 2019. He was also former Chief Scientific Officer in Exact Diagnostic Private Limited from 2019 to 2022, and currently he is serving as Director Research of Exact Diagnostic. Dr. Sangith has acquired extensive experience both in industry and academic research and has gained excellent reputation in creativity and innovation. Today's webinar holds special significance as we delve in the practical aspects of bridging the gap between scientific innovation and commercial success. Our distinguished alumni speaker, who has traversed this path with determination and vision, will share his experience, challenges, and triumphs, providing invaluable lessons for our aspiring young minds. For our students joining us today, the webinar serves as a beacon of knowledge and inspiration as we embark on your professional journey. Without further ado, let us commence this webinar with open minds and eager hearts, ready to explore the fascinating realm of commercialization in science. Thank you all for being here, and let's make this webinar an enriching and transformative experience for everyone involved. I encourage all of you to actively participate and ask questions and engage with our speakers. Thank you. And now, Dr. Nikhil Sangith, please.
Yes.
Yeah. So you can start your webinar. Welcome, and you.
Okay. Thank you so much. Can you see my slides on the screen?
Yes, sir.
Yeah. So thank you very much for inviting me once again to this lecture series. The first time I had this opportunity in the year 2021, and I am very thankful to HBNI for inviting me again. So I will start right away. As you can see, for the title, it mentions the word musings, which means reflections or contemplation of a scientist where I could connect innovative ideas and start commercialization in a startup environment. So basically, I would start by having a brief introduction of what my background is, of what kind of research I have done. And after, with a very quick brief introduction, I will move on to the second part of the talk, which will again take 3 to 5 minutes only, which will describe about certain important concepts which we need to understand in business, very fundamental terms which have no complications. So that you can follow that throughout my work, which I am going to explain through my journey at Exact Diagnostic Private Limited. So I'll quickly move into my background, what I had done through my PhD postdoc and as a scientist at Anna University. So as mentioned by Madam, I had completed my PhD at ACTREC, Advanced Center for Treatment Research and Education in Cancer, which is under HBNI, under Dr. Prasanna Venkatraman, who is currently the director of ACTREC. And we worked on structural biology, understanding the structure function correlation of proteins, and its degradation, protein degradation in mammalian cells, and its correlation with cancer. So, with this background, after 2015, the completion of my PhD, I moved on to a short postdoctoral stint at TIFR Mumbai, where we studied mechanical properties of proteins through atomic force microscopy in the Department of Chemical Sciences. After which, I moved to Anna University as a DBT scientist under the Builder program, where I worked under Professor K. Shankaran for 3 years from 2016 and 2019, where the major focus was on bacterial infectious diseases, its fundamental host pathogen interaction, and also trying to develop novel, cost effective diagnostic platforms. So, with this varied background, after 2019, I happened to meet Dr. Pavan Asalapuram, who is an alumni of Anna University. And he was a founder of a couple of companies in Sweden. And after a brief discussion with Professor Shankaran also, we decided to experimentally start a startup at Anna University itself, which was named as Exact Diagnostic Private Limited. So it all started as a single employee where I was the only employee there. I started as a CSO, and now we have a small team, and we are currently going on. So I will explain what we are doing at Exact Diagnostic Private Limited. But before that, I would just want to start how some small terms which we need to understand in commercialization before we go into the science part and how we went ahead with our work to commercialize. So, for all the students, I will keep my presentation as simple as possible. And for those students who have attended my lecture in 2021, may feel that it is slightly repetitive in the initial part, but because we have extended our work quite a bit, it will those things will be new. But to refresh your memory, I just want to describe a few terminologies which you will be able to follow as I go by my lecture. So basically, this is an era of many startups coming up, and what we should practically understand the difference between a normal company and a startup is that a normal company will have a very structured environment where you have a proper hierarchy of a CEO and then corresponding managers and then other employees down the line, and it is very structured with a lot of manpower, sufficient funding, and also there is a defined target for you to achieve, and the problems there are much different that what you see in a startup. In a startup where everything starts from the scratch, and it all ends up in to one or two individuals who are the founders, and there is a less manpower, and what we target may or may not work, and there is no particular job description for any individual. We have to be ready to do. So you can see in this there are so many different departments here where maybe one or two people have to do all the things initially till the company gets into a structured aspect and we enter into proper commercialization. So the challenges in a startup are completely different, where even there is low manpower as well as investment, with which we need to be very frugal at the same time and enable proper commercialization and revenue generation. So, how do you begin a startup? So, there are three basic ways to simply understand. Because there is nothing, because you are a scientist or you are from a art background or from a science background or a commerce background, startup is to start a small company. What you really essentially need is the expertise. or in a particular field, whichever field it may be. So if you have the expertise, you start looking into the problems in the society or in the field of research which you are in. Once you identify a gap, that okay, with my expertise I can address this problem, you generate the idea. So once you generate the idea for that particular gap or particular problem, you then develop the solution, for which they call the minimum solution. That you need not polish, and it need not work 100%, but if you can bring it to at least 70 to 80%, that we call it as the MVP, or minimum viable product. So if you are able to bring a product to that stage, that product can be tested, validated, and given to the customer, and then you can understand that that can give you revenue. So this is what the first approach. The second approach is, you may have a lot of expertise of being in multiple research centers as a postdoc, and as a scientist, as an academician, and you would have done a lot of research, and you would have a lot of information which may be solutions, ready-made solutions of the problem. So when you suddenly start seeing the problem, you may think that I have the solution already, because the vast expertise you already have. So you can fit in that solution and immediately go for the commercialization. So the second thing is a little rarer, but the first thing is generally the most common way, where they identify the problem, and then based on your expertise, if you can address the solution, you go into commercialization. So the third one is a very direct way of making revenue. If you have if you have an expertise, you can provide services to many clients who will come to you with their problems, and if you are smart enough and if you are efficient enough in bringing solutions to them, that would also generate revenue. So this is the three ways. You can do all three ways in a startup, or you can do a combination of two, or you can only follow one strategy. It is all up to you. So this is the basic thing. So it is not enough that you have a minimal viable product. There are three important things you should have after that. So the first thing is to validate whatever you are providing as a solution should be validated in your lab, or whatever method that has to be done in the proper way that is accepted by the regulatory authorities. Once that is validated, it can go into the market. But when it goes into the market, there are big players already in the market which are going to do a similar job. So your product should also have a USP. That is a unique selling point. That stands out. That is, it outshines the existing product in some way or the other. It may not be in a very big way, but it should be better than the current uh, than the than the existing product in the market. So this is called the unique selling point. So once you validate, you should also ensure, when you are doing all this, that it has a unique selling point. And then you should know your market, who are your customers, and where you can sell the product, and how much you can get revenue. So these are the two basic slides I wanted to introduce, so that I'm not going to I discuss any more business and bore you. But these are the two important things which we need to understand. Address the gap, find a solution, make an MVP or minimal viable product, validate it, and that should have a USP, and you should have a clear market goal. So now I will explain what we were doing in Exact, and based on these points, so that you can connect the business as well as the science, and follow the lecture, and clearly understand in simple terms. So in 2019, when we joined, Dr. Pawan Asalapuram, who is our founder and CEO, currently also the chairman of the company, he has also founded a company called Apirace in Sweden. So so we didn't know what to start with. Actually, it was an experimental basis. They said, we decided, okay, we will start. We will see how it goes for 6 months, and then decide from there on. So the first project which we were given was from this company. This is a services base. That is, they have come with us with a problem, and we have to solve and give it to them, and we will get paid for it. So this was our first project. So this was based on an enzyme called apyrase. As you can see here, APYRASE. So as you know, enzymes are catalysts that convert a substrate to a product. So the substrate are these four items, which are called nucleoside triphosphates: ATP, GTP, CTP, TTP, and that gets converted to AMP, which is the product: GMP, CMP, TMP. So this was the enzyme which they had, and they were testing this enzyme in a diagnostic platform in urine. And unfortunately for them, in the urine, the enzyme was not working. So they came to us saying that you have to find a solution, hook or crook, make the enzyme work in urine. So I asked them suggestions, because they have been working on this enzyme for a couple of years, and I asked them what they think may be the reason. So they told there is a lot of salt in the urine there, so that may be inhibiting the enzyme. So you mutate the enzyme with your in gut feeling, what you are feeling, and you show you if you solve, we will pay the money for your company. I didn't agree with the concept entirely until and unless I do the experiment myself. So we purified the enzyme, and then we did the assays. That is, we checked the activity of that apyrase or API1, which is the commercial name. We checked the activity in urine. But what we found surprisingly was that enzyme, it had nothing to do with the enzyme. The components of urine were actually destroying the substrate. So the people who were complaining about this were obsessed with the enzyme, saying that enzyme is getting inhibited, so this is what is important. We have to clearly see what is happening in the experiment, and once you understand the root cause, we can address the solution. So when I came to understand that the urine actually destroys the substrate, which is ATP or TTP or GTP or whatever it is, we needed to focus on protecting the substrate. If I protect the substrate, the reaction would go forward. That is what I understood. And therefore, we developed a unique method, which I cannot reveal, unfortunately. So this is the composition of urine. We tested every component of urine that can inhibit. As you can see on the right, the blue is the normal method they used to do in Sweden, where when they add the components of the urine, it gets inhibited. And we at Exact actually developed a method that could overcome this inhibition, where you completely negate the effect of urine on the substrate, and therefore the enzyme is able to act on the substrate, and therefore the reaction occurs. So this was the report which we gave within 2 months. And then they were so happy, and this was our first revenue. So this is a I always in my lecture start with this experiment, because the first experiment or the first project is always close to your heart for the company. So this was the first project which we got recognition, and we got some revenue. And then we were again back to square one, that we what to do next. After 2 to 3 months, what should we do? They gave us a deadline of 6 months, but we finished it. So now what should we do? So I thought that we have API1 and we have an agreement with apyrase. So why can't I work more on API1 and use this as a minimal viable product? Please remember my a few two business slides. Can I modify, improve this product and make it to another useful product which will be commercially viable? So what with my training at Actrec as a structural biologist in Dr. Prasanna's lab, I, I was taught to look at the structure in different ways and predict mutations. That is, which amino acid or which chemically, how can you alter the protein so that you can change the activity of the protein, either increase or decrease. So I kept changing different residues or the different amino acids on the protein, and then I converted this enzyme to another phosphatase. That is, it is a class of pyrophosphatase, but I converted it to a phosphatase where its specificity entirely changed to different other substrates that were very important in the biochemical industry. So these are the two new substrates that could get hydrolyzed, and this is the new version of the enzyme which we call API2. So API1 was my minimal viable product, and I mutated it and made it API2, which can hydrolyze two different substrates, and this became another product. And you can see here API1 can only hydrolyze ATP, where API2 can hydrolyze TPP and PNPP, which are the two new substrates, thymine pyrophosphate and PNPP, paranitrophenyl phosphate. These substrates are utilized in the biochemical industry for an enzyme called alkaline phosphatase, where they don't prefer this enzyme because of the high pH cofactor that is required, and inhibitors like EDTA affect the enzymes. But with the case of API2, all these drawbacks are negated because it is resistant to inhibitors, it does not require cofactors, and it can even work in water. So this is the uniqueness or the USP, as I told, the unique selling point of the API2 enzyme, which will have the advantage over alkaline phosphatase. Above all, API2 has a higher affinity to the substrate. So all these factors, when we informed Apires, they were again happy, and we had the next, payment done because of this service, and we patented this. And as an inventor also, I'm fortunate to be part of the patent. So and the patent has been filed, accepted in different countries as well. So now as I said, we have one MVP, and then we converted it to another product. Now we have two products. Now similarly, when our discussions always go with the CEO, he was stressing about biofilm, because after post COVID, the disinfectant market actually boomed. So people were worried about biofilms, how to remove biofilms, especially in the hospitals. And it is you can see the market is huge, especially in Western Europe. So I came across the fact that biofilms are actually formed because of ATP, GTP, CTP, TTP, which are substrates of API1, which I showed you in the first project. So if I use API1, I can at least remove the precursors for biofilm. But the challenge was, biofilms occur on surface of the table, on surface of instruments, where the cleanliness is not maintained, and bacteria actually signal each other to form a layer, as you can see on the right, and they become resistant to antibiotics. So I should find a way that my enzyme should be engineered in such a way that it get others or sticks to the surface, and that is how we engineered this enzyme by lipidating it, but through a protein engineering technology, which again I was trained in this technology by Professor Shankaran at Anna University during the 3 years scientist tenure, where this expertise helped me to engineer this enzyme. And therefore you could actually adhere it to the surface. When you coat a few nanograms of enzyme on the surface, you can I make this enzyme, you can find that on the after wiping the surface, you find that the enzyme is attached to the surface, and you can see the activity. So based on this, we developed a reagent called API1 biofilm slayer that prevents the formation of biofilm. As you can see in our validation experiments, different bacteria that are known to form biofilms as very threatening biofilms like Salmonella, Pseudomonas, uropathogenic E. coli, we have shown that that our product developed engineered with this engineered enzyme prevents the formation of biofilms. So again, an MVP improvement. Minimal viable product AP1 is engineered as a lipidated one to address a different problem, and therefore you have the third product. So this is the way you can improve one product and get multiple products derivatives, so that your business and ability to commercialize also expands. So as I so now I will move away from apires and all the, we are going to another set of products which actually happened to be discovered by accident. So serendipity is an occasional event during science, where you accidentally you are doing something, and then you happen to discover something else is happening. So actually, I was in a working on a services project for another pharma company, where they had given me to assay an enzyme and stabilize its product. So I used to use many additives, many additive mixes to stabilize the product. I used to set up the assay in 12 well plate, and but due to lack of resources, I used to reuse the plate. I used to wash the plate. I didn't never used to throw. It was not use and throw because of the initial phase where I am very stringent in spending money during the initial phases. So I used to these kind of crude experiments I used to do by washing the plate. So to make you understand how this discovery happened, I will explain it pictorially, where I use this a 12 well plate, and I was doing an experiment where I used to add an additive mix in different ratios with enzyme and reactants. So for example, I had put it in the first well. I finished the experiment, then I threw all the things, I washed it, then I start the next experiment with the same enzyme on the same plate, but I don't add the enzyme in the first well. I only add it as a control well, where I only add the reactants. I don't add the enzyme at all. But still the assay was working. But I was wondering that I made a mistake by adding the enzyme accidentally. But I again threw all the reagents, used the same plate, washed again, and then repeated the experiment and add only the reactants in the first well. But again the assay worked. So that means something, when I was doing with the additive mix, mixing different formulation, something is enabling the enzyme to coat on the well. So I went back to the additive mixes, in different permutation combination, and I found that I could develop a reagent which we call it as a insta-coat buffer, that instantly coats any protein, antigen, antibody, enzyme, whatever you want, on a plastic plate within 10 minutes to 1 hour. So, with the multiple validations with different enzymes, proteins, we established the fact that we had the right formulation, which we call as the exact Instacoat buffer. So, what this exact Instacoat buffer does is that in different biochemical assays, especially ELISA, which is the most commonly gold standard immunoassay, which is currently used in laboratories in all biotech labs, as well as biochemistry labs and diagnostic labs. People coat antigen or antibody on plastic plate, and the conventional method that is used, they use either PBS or carbonate buffer that takes 6 to 16 hours for coating. Whereas now we have a formulation which will immediately coat within 10 minutes to 1 hour, depending on the protein. So you don't need to waste time. As you can see our results here, you can see that the enzyme is coated on the plate. We used different brands of plates. So there is extensive validation that, we are not showing that, okay, in one brand of plate it was. Different brands of plastic, ranging from high quality to the locally made manufacturers also. So you can see that the exact Instacoat outperforms the conventional carbonate buffer and it does not affect the function of the protein. You can clearly quote, and this is one of the first steps for ELISA, and that is the rate limiting step that takes a long time for coating, generally efficient coating. So we even compared different brands. You see the carbonate buffer 6 hours you have to incubate. You see a variability in coating from different brands. You see highest brand is performing very well, but low cost plates are not performing up to the mark. Whereas when you use our formulation InstaCoat, it can uniformly coat across different irrespective of the brands, and we have given multiple samples to different institutes, including Center for Drug Research Institute, CSIR, CDRI, where we have got the feedback that it is working. We have started selling this buffer also. So this became a very good product for us, and we have filed the patent also for this product. So what can we do with now? We have the XIC or exact Instacoat buffer, instantly coating. But what all, like this is my MVP right now, like API one. What all can I do from this is what was my next question. So once what I did was, the first thing was the university was close to me, and I was associate. I had a few friends who are lecturers in the university. So I used to go there and ask them what kind of practicals they do. So they told me that they are buying ELISA kits, commercial ELISA kits, teaching kits to teach the students from big companies like Bangalore Genie, High Media, paying a lot of money for just 4 experiments, 3 experiments. And I went through their protocol. All of them had a 2-day protocol where the coating is done overnight. Next day, first day, students are called, they do the coating, then they go home. The next day they come, then they do the ELISA experiments. So 2-day practical class goes. So why I thought, why don't we make kits so that everything finishes within 2.5 to 3 hours, so that it will be ideal for the teachers to conduct a practical class in all educational institutions. So this was one thing which will also validate my product, which is exact Instaco. So in a startup, we have to be hustlers. That is, we have to get the thing done, hook or crook. I mean, in a legal way. So we started looking out for sources where we can cheaply resource components for the ELISA kit, and we actually compiled and developed 2 teaching kits for the students. And as you can see, within instead of a 2-day protocol, we have everything done within 3 hours. And you can see the conventional buffer is outperformed by our exact Instacode buffer. And this we did a demo in the university, and then the at the university actually recommended for ordering this. We have supplied the orders, and we have got good recommendations and feedback even from the director of the institute. So this was quite encouraging for us, and we have now expanding our sales through other states also. Now right now only given to Tamil Nadu, and our exact Instacode has also been, I mean, we are also selling it in other places also. So this was a very good validation for us. It is not only a validation, this becomes a product for us that gives us money. So in both ways, it is a win-win situation. And in our kit, instead of 4 experiments or 3 experiments, we give 10 experiments at a very nominal cost, and your time of the experiment is also low. So in all ways, it is a win-win situation for both the customer as well as for us to make profit. So this is the next product which we developed from exact Instacode. Now you can always say, oh, this is just a teaching kit. We have not entered into diagnostics. So diagnostics is a very big field which needs a lot of validation. So we wanted to enter it, and we were brooding on the idea. So I always wanted to develop with whatever we have the lipidation technology which I have learned from Anna University, and the protein engineering and other biochemistry knowledge from my PhD, and also the experience till now. We wanted to develop a platform technology which for the ELISA, which can be used for multiple diseases, disease detection. So the preference for any customer will be a high sensitive assay, because the lesser the amount of product I can detect, the more successful and more sensitive is my product. So that should be high. At the same time, I should not give an expensive technology to the customer. They will not buy. At the same time, it should not take much of the customer's time also. So it should be rapid, it should be affordable, and it should be reliable in terms of sensitivity also. So that is why we named our ELISA as a rare ELISA, called Rapid, Affordable, Reliable ELISA Platform. So what should now the next thing was, what are the components of this ELISA which we have already developed. So the I have already talked about the exact insta code, but the high sensitivity part is brought in by a concept of lipidation, which is adding a lipid moiety to the antigen or antibody which we are going for the doing the immunoassay. So when we are lipidating the antibody or antigen, what happens is that there is it ensures a proper orientation of the antigen on the plastic ELISA plate. So this orientation enables maximal interaction and facilitates anchorage also, and it enhances the sensitivity of detection on the plate. So this lipidation technology, which we have developed and patented, can be applied to increase the sensitivity and to reduce the cost and time. You have exact insta code as the next component, which will rapidly code, so the time is less, and also on low cost ELISA plates where the cost is even brought down. So on one side you have high sensitivity, on the other side you have low cost and rapid assay. So you can name this as a rapid, affordable, reliable ELISA. So I have to prove. I proved with respect to InstaCode. I have shown you the data. I have shown you the feedback from the customer, from other products also. But what about lipidation? How does lipidation increase the sensitivity? So I should have a model system to show that lipidation increases the sensitivity. So we chose a disease in shrimps. Shrimp is a aquatic organism which actually is a big market where there are many shrimp farms across the country where they cultivate shrimps and then export. Unfortunately for the shrimps exporters, this virus called white spot syndrome virus infects and destroys the shrimps and kills the shrimps. So they cannot cultivate it and it spreads also. So to detect that, they are using immunoassays. So there is a huge impact in this industry. So we decided to develop an ELISA first as a test model, not to the market, but to convince ourselves with the help by lipidating the antigen. So we chose an antigen called ICP11, its native form, and with our technology, we lipidated it also, and we had a lipidated LICP11. So we wanted to see which is more efficient, lipidated or the native form. So according to our theory, lipidation should increase the sensitivity. So first we saw the adsorption of the lipidated versus the native on the plastic plate, and you can see clearly when you put on the first well itself, almost 90% of the lipidated antigen gets adsorbed. So lipidation enhances the hydrophobic interaction and therefore enhances the adsorption. And on the right you can clearly see at low amount of lipidated antigen you still get a very good detection ability compared to the native NICP11. This clearly shows that lipidation increases the sensitivity of detection. Not only in this kind of an assay, we also diluted the antibody to detect the lipidated antigen. You can go to very high dilution to still detect the lipidated antigen compared to the native, which cannot be detected at a high dilution. So this clearly shows that when you lipidate the antigen or antibody, the sensitivity of the assay increases remarkably. So on one hand, we have high sensitivity, on the other hand, low cost, rapid. So our rare ELISA platform is set. Now where can I pitch in? Which diseases should I address? Now I have to search for the gap. Now I have a technology. Now I have to search for the gap where I can really address it. Now we don't have the muscle power or money power to have regulatory, clear the regulatory bodies at this stage for human diseases. So I started writing to veterinary institutes. I was trying to contact Tanuvas, which is Tamil Nadu Animal Science University, which is one of the best state universities for veterinary sciences in India. And they have a very good faculty as well as a very good infrastructure also. So when I wrote to them that I want to meet the director, he gave me an appointment, and I met the director, Dr. C. Soundararajan, and he was very happy to collaborate. So I asked him, with on this kind of a platform, what kind of disease can we test? So he said that you develop a diagnostic kit for helminthic parasites. Helminthic means worms that infect cattle and goats. He said this has a very large socio-economic cost because it infects these worms, create symptoms. These symptoms are very common, and you cannot identify the disease because the symptoms are very common. The medicine for these worms are available in the market, which are very cheap. 15-20 rupees you will get the medicine, but to identify which worm is infected is the most difficult thing. If we and there is no current diagnostic test available, so you develop this for the detecting these worms. So these worms also cause a huge loss to farmers. According to the current report in 2017, in a state like Uttarakhand, almost 90 crore loss has occurred only due to the death of cattle due to the worm called fasciola. So I have chosen three different worms which are distributed both in plains as well as hilly areas, so that different I can test the efficiency of my kit with the different antigens, and then decide what kind of commercial kit I can finally for commercialization. So at the validation stage, we chose three worms. So why did we choose ELISA for this? We can do RT-PCR also, but that is the most important gap which we have identified here. We cannot do RT-PCR for these three worms because these worms infect the cattle or goat, and these worms, the disease is caused by immature worms. They don't even lay eggs for 1 to 3 months. So there is a pre-patent or latent period where the symptoms may be there, symptoms are mild, but you can't detect the disease. You cannot detect by PCR because there is no DNA available in the blood. You have to slaughter the animal and open the liver or intestine to see whether the worm is there. But antibodies will be produced in the sera against the worm. So immunoassay is the only way you can detect this. So this becomes our advantage for these worms, and you need very high sensitivity for this. So we have a high sensitivity platform, we have a fast platform, and we have a robust platform. So we decided to partner, and Dr. Soundararajan sir was kind enough to introduce me to the vice chancellor of Tanuvas, where I pitched the idea for 3 minutes, and then they accepted and we signed the MOU. With that pitch, actually we applied, and we simultaneously we also signed an agreement with the ICAR National Meat Research Institute as well. With so one of my team member, Mr. Uday, is also a great hustler and enabled this agreement to come into being. And now we are into the validation stage. We are going into that. But with this idea, we pitched to the government of India citing the new technology, the rapid technology, as well as it is addressing a socio-economic cause, where we are for the first time we are trying to develop it for the veterinarians for diagnosing these kind of diseases. And we have been awarded the biotechnology ignition grant from BIRAC, that is sponsored by DBT, of rupees 50 lakhs. And we are now just 6 months since we have been granted the money. And we have started the work by we have identified the immunodominant antigens, and we have optimized the rare ELISA, and we are now trying to go into the field to with the veterinarians. So the process is underway. So all those data I am not presenting, but this is how one exact insta coat and our lipidation technology, how it has how we could convert to product by product is based on what USP we identify, what gap we are identifying, and how we can generate revenue both in terms of selling the product as well as in proposing important projects which are of long term vision and which can get us grants that can support us to do those projects and develop the products. So this is the ELISA platform. So I will go for couple of more products, and then I will close my lecture with a few points. So during so this is again we developed another enzyme which is a thermostable DNA ligase. We are also quite good in producing enzymes at our facility, and we my training is also in enzymology also. So my chairman was actually is also a founder of another company called MP Diagnostics in Sweden, where they use this enzyme. So they were purchasing at a very high cost from Poland, and he asked me whether we could manufacture this at a very low cost, and we could do that. So there is no very big difference from what is available in the market that my enzyme is better than that. No, I don't think so. But at least at the cost point of view, it is much more affordable. So we have done that. So that is also one of our product portfolios. And then the last product which I will be discussing as of today is the another reagent which is called a new pro cleanser. New stands for nucleotide, pro stands for protein. This is an all-in-one solution to remove nucleotide and protein contamination for PCR workstations. So these products we developed with different formulation, where this came out of a discussion where again my CEO in Sweden was telling me that MP Diagnostics is buying these solutions which are sold by Thermo or other big companies, and they are paying around 10,000 rupees for 200 ml. And they are buying liters and liters and spending huge amount of money, especially corporate companies. So he wanted me to develop DNA. You have one solution, RNAs you have one solution. He said, can you develop a solution for all three? DNA, RNA, protein, everything should go, and it should be cheap, and it should not be costly. It should go within 1 or 2 minutes. So we did, we formulated, and we have DNA, the nucleotides also disappear. qPCR also showed us that even minor detectable DNA is also not there on surfaces by swabbing, and also protein also gets destroyed. So from all these things, what we have for all the products I have just explained, based on the principle of identifying the gap, validating, and extending the innovative idea to multiple products. We have patented, these are the patents which has been granted at different patents for XIC, API2 and API1. uh, and and now this is the exact diagnostics portfolio. On the left you can see the products which we have developed from scratch, making one by one, taking one minimal viable product, developing the next based on what the customer needs. So for me there was no pre-planned way of doing things. whatever situation I was there, whatever infrastructure I was given, whatever minimal facility was given, we whatever we could utilize, whatever we could beg and borrow from our colleagues and collaborators, whatever help we could get from our advisors like Dr. C. Soundarajan from NMRI, all the support. We were able to do these products, and the long-term projects you are seeing on the right are the ones which we that is our vision, to bring unique products which will create a bigger impact for the customers in this country. And in the center you can see the platform technology, the lipidation technology, and also the rapid, affordable, reliable ELISA which we have developed. And we are providing services also for those people who have problems. We charge nominally and we do the work for them. So these are the ways exact generates revenue, and these are the plans that exact currently has. So the journey is continuing, and I just want you just recollecting how we started. In 2019, it was a single employee that I was the only person, and this was the room where API2 was developed. I just had a table and chair, and and Anna University was very kind enough for not charging rent for 2 years. They just allowed me to work in a corner of a lab, Professor Shankaran's old lab, where they gave me a corner, and that was the only thing I had. So I collaborated with different professors at Anna University for their work, their publication, their patent, and I am also part of that. They have given me credit for that too. But where my contribution there, I asked in exchange their facility, because we didn't have even a proper freezer, a proper fridge at that time. And slowly, when money started pouring, because it is very difficult to gain trust when you are claiming these kind of ideas. Oh, this is an Insta code. This is this. People will not believe you, and even your CEO will take time to believe you. So it takes a lot of time to get the trust. And once the CEO trusts in you, so this trust took 2 years, and that is why he offered me stake in the company, and also provided me with the directorship, and enabled me to take independent decisions from there on. And from that, and from the funding we have, and also from the revenue we are generating, and how our CEO is supporting from his investment, we have now a 200 square feet independent facility with a few of our own instruments, where we at the Anna University Bio Incubator, and we are now trying to expand our team in a different city also, where we can start manufacturing, go into the regulatory aspects, and go build a better team. I mean, better team in the sense of larger team, so that responsibilities can be shared, and more amount of work can be done, and therefore more products can be produced and sold, and therefore more revenue to keep both the company as well as the customers happy. So I should thank the entire team, without whom I think I won't be talking today. Actually, Dr. I should thank Pawan for trusting me first, because there was a time in 2019, but nobody was willing to believe that we can do something new, something new. Pawan actually trusted that, and slowly, gave me that space and freedom to explore. That is very important, and not imposing only his ideas. And of course, Dr. Shankaran, who gave his lab some of his resources, and also when money was required, also sometimes he came in, and that is and his guidance is always uh, important in our experiments also. And also Mr. Raghavendra Gaud, who was in GE Healthcare for almost 25 years as a Asia Pacific managing director, and now is as helping many startups. His motivation and his way of looking at business actually changed our way of looking at the things. And I have also learned a lot of things, especially in terms of business, from him. And of course, the workforce actually comes from these two people. Navya, who is now helping me with all the experiments in the lab, and also manages the lab, along with Uday, who is the assistant manager for regulatory affairs and quality analysis. He is a very good hustler, getting to get things done. And actually, without this support, I think all this work would not have come, and I won't be talking to you today. So the journey continues. As the title suggested, these are just incremental steps. We have just started. In the earlier lecture, I said that the baby was just born, it is crawling. I think now we have started to stand up, and now we are just starting to walk. The baby still has to grow, and I hope the company grows. And company B's should be an instrument to solve major problems and concerns that bother the society, and also keep the customers happy, so that revenue comes and the company is always happy. So I thank HBNI and Actrec actually for everything. HBNI from PhD days, and also for allowing me to speak to all of you again. And Actrec, of course, I cannot forget those 6 years or 7 years which I have spent as a PhD student in Dr. Prasanna's lab. And I think the contribution Actrec has given in my life is humongous. The amount of learning that has happened in that period, I think this is a moment I should again reiterate the fact that without that fundamental knowledge and training, I think that gave me the confidence to face all the challenges that we faced in the startup. So I thank Dr. Prasanna, as well as I thank all my seniors and my juniors, my batchmates, and all the Actrec faculty, including my doctoral committee members, who have always been a guiding force at that time and have contributed to whatever I know. Thank you very much. This is my email ID, and this is my personal number. You can reach me anytime. All the students who wish to come to a startup environment can contact me. And I want to tell the students one thing, that don't look at the facility, look at the idea, and look at how you can improve the idea and how you can contribute. One person, two people who have inspired me, out of which one is Jack Ma, and his famous 4 sentence saying is very important. At the age of 25, we have to go to a boss where we have to do multiple things and we learn as much as possible, because at the age of 25 to 30 is where we can work hard, we can run around. 30 to 40 also we can run around, but at 30 to 40, with the expertise, you have to try multiple things and find out where you are good at. At 40 to 50, we have to do only what we are good at. And 50 to 60, we guide the next generation so that they become proficient. And after 60, it is your choice. You can be with your grandchildren, or continue to work for in whichever way you want. But at this age span of 25 to 30, I want you people to see the idea. The money factor will follow. Money is very important. Without money, where money is important, money only can work there. But at the same time, uh, be passionate about what you do, and follow that with discipline and focus. That's all. Thank you so much. I will take questions if everything is over. Thank you. Thank you.
Thank you, Dr. Nikhil. I think
Yeah.
before we go for the questions, I would like to congratulate you for
Thank you,
this
sir.
excellent journey that you have made.
Thank you.
Starting with a PhD scholar to till today, and I'm very proud. And you are a role model to all the students in the life science community. I would say specifically that you started with relying on your own ideas, hard work, persistence, and creativity and innovation. I think you succeeded starting with a small space with a table chair. Today you have your own team and your own equipments and office and more ideas. And now this experience, what you have built over the years, is your strength. I am sure that not only that you have shown the way to others, but I am sure that you will also take some of the students. See, we may have some 200 students in life science, but I will be very proud even if some 4 or 5 students are interested in having their own startup, having their own journey. You should mentor them and you should
Sure, sir.
make an open call. You have given your email ID, you have given your number. And any student, I am not interested in crowd pulling, I don't want 100 students to attend a talk. I want even 10 students mean they mean business. They're interested in doing something. Everybody need not go for, behind some company for a job and some fellowship somewhere for some time and then look at other person for the job. This is what a very famous quote is that you should not be a job seeker, you should be a job provider. Today, I'm very proud that you are a job provider. If you want on your project, you can hire people, maybe at an assistant level, you must be started doing it. Around the time I'm seeing after 10 years, you will be one of the leading person and you may be providing job for many. This is what is going to be the destiny and God bless you. And I wish you all the best in all your
Thank you.
endeavors. Thank
Thank
you.
you so much. Very honored. Thank you, sir.
Thank you.
Question.
Thank you, Nikhil.
Thank you. Thank you.
The question, there's, yeah,
There is a question.
there are question in the this YouTube or that is in
So, madam, I think you are open. Yeah, you can open. You are unmute and put up.
you. Appanagal. Yeah.
Yes, hello. Am I audible?
Yeah.
Hello,
Yeah,
sir.
you are audible.
Thank you for the presentation. I have a two-part question. One thing, you were talking about the beginning journey of the startup. So I was wondering, how do you go about market research, and if there are ways in which we can use existing data for our startup beginning? Thank you, sir.
The simplest way is to contact vendors. I mean, there are different, I believe in organic interaction. Because personally, when you are directly connected with people, you get a feel of what the customer feels. Of course, market data is available in terms of numbers. You can always go through that. There is no problem with that. That is always available online, so you can go through. But you have to see what kind of brands, you are, addressing one particular product. What are the other brands? Big players are there. Like you take Thermo, you take Sigma. They are the big players who have captured a major share of the market. Correct. So you should see how much that product is being bought by the customer. So you are in a scientific lab setup. You are having many distributors visiting you for their sales, where from you are buying your chemicals from. So when you start interacting with them, they will tell you they are the best people because they are selling it directly. They know which product is going, which product is not going. So when you understand that kind of organic way of understanding, will play in a very much better way for you to understand what is going on in the market. Data is always there online, where you can always download and look at the data. That is not at all a problem. This is how I do my market research. But it is always, because we are science people, we should always have a co-founder or somebody in the company who look at that market uh, in a commercial way. So everything cannot be done by the single person. So I do it organically, and then the professional way is taken by somebody like my chairman, who is not doing much science in the lab, but he is going around meeting people. So you need a founder like that, co-founder like that, who does that. Yeah.
Okay, thank you, sir.
Yeah.
Thank you. Any other question?
Somebody in the chat box has written something.
Yeah. That will. Before.
Put the mic on.
I have many questions. One will be technical and the other maybe non-technical.
Yes.
While going through your talk, you mentioned about biofilms and how your products and how your ideas try to tackle it.
Yes.
Now, there is one more thing which I got to know. Not coming from a medical background, this may sound a little primitive, but I've heard about something known as hospital-induced infections or diseases. So,
Yes.
how? I mean, is there anything that your company or your ideas are looking for to tackle those things? Because it is something which people are getting after going into an hospital or in the intensive care unit where they are supposed to get treated well. So
Correct.
is there anywhere in your range of products or in your spectrum wherein you are trying to address this thing? Because uh, if biofilm can be tackled in a way, then these things may also be looked into.
Yes. So, yeah, so the question is valid where hospital-induced infections are quite common these days. And, especially an organism like Staph aureus, they say MRSA, methicillin-resistant Staph aureus, all this, spreads to different patients. This occurs because, they of certain negligence or sometimes proper cleanliness or sanitization is not done up to the mark. So these organisms are actually very resistant to certain antibiotics, therefore they manage to survive even sometimes when we clean it. So that is why products like biofilm cleanser will play a major role, because the what is targeted is not killing the bacteria alone. What is targeted is to eliminate the precursors of those which form the biofilm. So that is why we are trying to introduce this into hospitals right now. But there has to be a proper. I mean, they are actually validating whether it is fit for that. So that is going on. But as a laboratory cleanser, this especially in diagnostic labs where they have a lot of bacteria, this is quite useful. But as you said, for hospitals, this can be the product itself can be a solution, is what is I am telling you, because we are targeting the precursors of biofilm formation, not only killing the organism.
Thank you, sir. There are one or two other questions. First one is I would like to know where your company is located specifically.
Yeah.
Uh,
Yeah, we are located actually in Chennai, at Taramani campus of Anna University. So Anna University has a small bio incubator uh, where around 8 or 9 companies are there, small startups. So we are now incubated there, where the R&D and all these experiments are going on. We also have, I mean, we are operating in Hyderabad also, but not as an office only, but not as a R&D unit. So, yeah.
the reason for asking this is that in case if we try and let's say plan a visit, like you invited us gracefully that the people from HBNI or Attract or people who are connected with HBNI can come and have a look
Sure,
at your startup, how they work and what is the environment over there. So the reason for asking this is if we think of visiting or if in a way uh, we come for some internship opportunities also. We would like
yeah.
to know where this
The
startup is
location will be
located.
in Chennai. Chennai.
Okay.
Yeah. The operating unit is in Chennai. Yeah.
Okay. Thank you.
Yes, Nikhil, there is a
Yes.
question in the chat box. You can
Yes.
also read. It's from
Yes.
Divya Gohil. That is, what
Yes.
is your advice to students thinking to transition to industry after their PhD. Specifically, I would like you to share your opinion on doing a postdoctorate before entering the industry. So what is your views on that?
See, postdoc always enhances your skill set, but it becomes more focused. So learning is always helpful. I don't think postdoc will be a hindrance to entering industry also. So if you are thinking of learning more, and if you find a lab where your skill set can improve in a particular thing, but you have to keep a deadline for that. That is not more than 3 years or not more than 5 years. So that aspect should be there. And yeah, the industry is a different. So if you want to, I mean, go into an industry which is already a corporate setup, you can always go there. You will have everything given to you, and you can work there. Uh, are you asking in terms of a startup or in terms of a postdoc? Is actually helpful. uh, there is no, and it is also, you can also have an opinion that postdoc is not necessary also. If your skill set is good at the end of your PhD, a postdoc is not necessary if your focus is only industry. So I always didn't want to do a long postdoc. It is my personal feeling was that I didn't want to do a lot of postdoc. I wanted to do something which I can translate. I can translate. So that is why I jumped to Anna University under Dr. Shankaram, because he was an academician but looking towards developing products for the industry. So that you have to search avenues like that for your postdoc. So if you are going to those kind of labs, you are molded for the industry, and then you can enter the industry. A
I think
startup is yes.
ultimately that how much skill set you have.
Yes.
Yeah, sometimes, even after MSc, some people
Yeah.
may be, getting into that if they feel they're confident they can do something.
Yes,
Now with
definitely.
PhD over 6-7 years, definitely
Yes,
you'll have your own skill set, but
definitely.
you have to have courage to enter into that.
Yes,
I
yes.
know that a lot of struggles one has to make. It is not that
Yes, yes.
easy. I
Yes,
mean,
yes.
today we can see smiling face of you, but
Yeah.
I know that it would have been much more difficult. You are
Yes,
not
yes.
talking anything about your personal friend, but
I'll tell you in a couple of minutes, sir. There you have seen 8 or 10 products. I mean 10 projects which I have seen. You see on the slide. There
yes.
are many which have not reached there. There are many where which are frozen, or we cannot take it forward. So there
Okay.
have been many failures. It is not
Okay.
that this is a cakewalk and you just come. It takes a lot of there is a lot of risk, honestly. There's even in academics there is a lot of risk. So I think we should be prepared to fail.
I think you
uh,
need three things. One
yeah,
is that your academic background.
yeah,
Second thing, you need somewhere, mentoring support with
yeah,
academics and research.
definitely.
And
Yeah,
third, I think the most
yeah,
important thing is that your family support. This is
definitely,
very important also.
definitely.
Somebody has to be behind you in your family that don't
Yeah,
worry about anything. You continue on this. I will support
yeah,
you. This
yeah.
is very important. I
Yeah,
think then only somebody.
in my case, I was fortunate that getting training from Shankaran sir in terms of product development. So that actually, and second thing, the trust that Pawan put in me after a period of 6 or 8 months. So financially, I was sound at one side. So I was not looking here and there. Neither, I just made a commitment to Pawan that I will not leave. So that ethical aspect is also there. So when
Yes.
somebody gives you, a founder puts everything in the company. He puts money. He's investing in you. So you should also give it back to him. If you have that in your heart, you will definitely get 1 or 2 products for him or her. So that I think students should have in their heart. Guidance you will get in your postdoc, and if you are applying for a postdoc, that is, if you want to go to industry, go to a lab where they do application based, where you are interested in, and then you can shift to the industry. But if somebody invests you, I would always tell you, you give them back twofold, threefold. They will look after you. I think that is what Pawan trusted me, and I also promised him that I will not leave him. Yeah.
Okay. Yes, madam.
Thank you. Thank you, Dr. Nikhil.
Thank you.
And thank you all of you. Thank you for a very, very, very wonderful lecture. And thank you for all you for your active participation. And thanks. And so we'll have a series of alumni monthly webinar, and we look forward all the alumni who can help in this and who can also guide. And I will ask, I will also request you to ask, request other alumni who can also give their insight about their experience. And so we'll.
Dr. Nikhil, you
Yes,
can
sir.
also, if you come across any other HPNI, successful, startup and the Make in India scheme,
Sure.
people who are known to you with HPNI background,
Sure.
so you please let us know so that we will ask their experience also to be shared. And
Sure.
additionally that even non-HPNI at your age group, and
Yeah.
like your contemporaries now
Yes. Yes.
in life sciences or any other area, please write
Yes.
to us so we can
Sure.
also call them sometime to address our students and mentor them.
Okay. Sir. Okay.
Okay. Thank you so much for sharing your
Sure, sir.
time.
I'll thank you for the opportunities. Thank you so much.
Thank you.
Thank you.